All right. Sorry, I'm trying to get it to full screen and it won't respond.
Okay.
I think part of the problem is I'm using Windows 7 and this equipment is not made for Windows 7. So sorry about that. But anyway, Ted did a great job of talking about the history of the salt marsh. So I won't go into that at all, but been working with the Salt Creek tiger beetle for a long time. And back in 2006, I thought a good thing to do would be a comprehensive insect survey, because as far as we could tell, we'd never been able to find any literature that documented the number of insects or the diversity of insects in the salt marsh, despite the fact that the university's been here so long. So we started in 2007, basically. We got some funding from Fish and Wildlife Service, some Section 6 endangered species funding. We ended the intensive survey in 2009, but basically kept collecting insects up until present time. The main study sites, Arbor Lake, Shoemaker Marsh, Capitol Beach, and Little Salt Fork Marsh Preserve, which was at that time, I think, a TNC property, Nature Conservancy property, and then Jackson, and then just a couple privately owned areas. We had already been collecting insects occasionally back when we started looking into the Salt Creek tiger beetle. So we've got records back to at least 1992. And what we did is we collected from April until October. So the collecting techniques we used, of course, a net. We had 3 different types of net: aquatic net, sweep net, and an aerial net, used for different type of habitat. And then we used a black light or a mercury vapor light, which we used with the generator, and we put it out there for several hours and basically collect everything that we could find. And then we bring it home and freeze it. Okay, Pitfall Trap was another technique we used. Pitfall Trap is basically a cup or a couple cups that are buried in the ground up to the edge or the lip of the cup, and then they're covered with like a plate or something like that, some kind of cover to keep the rain and such off. And then they're filled with the pink antifreeze, the safe antifreeze. Insects will stumble in or walk in to get away from the heat, or at nighttime they will just stumble in and fall into the antifreeze and die. And then the next day or a couple days later, we go and collect those, put them in alcohol or freeze them. And then we also used a Malaise trap. If you're not familiar with the Malaise trap, here's the exact trap that we use, purchased from BioQuip products, and it basically catches anything that flies in from any direction. This is about, probably 6 to 8 feet across each side. And then we had it actually touching the ground, so we'd get some of the crawling insects that would crawl up. They crawl up to the top, and then they go into a kind of a funnel and get trapped, and there's a killing agent inside the cup. And then we check on those every couple of weeks or so. Okay, so we collect these insects, we bring them back to the lab and we'd freeze them, and then we pin them at a later time. We tried to identify everything to species if it was possible, but a lot of the insects, there's just no keys known to species. A lot of these insects were some that had not been really studied much, and so maybe the keys were 50 or 100 years old, or just no keys existed. So tried to do the best we could and take it to the lowest level that we could. I sent quite a few insects out to different experts to have them identify ants, velvet ants, some small Hymenoptera wasps and such. Whoever I could find that was able to identify ground beetles was a big one. And then there's some families, such as some of the flies, that remain unidentified to species. Okay, so as of this year, 2,895 specimens have been databased. Now we caught a lot more than that, but some of the multiples we didn't even add to the database. We just had so many of the same thing. So we actually caught a lot more insects than that, but we caught 13 orders, 173 families, and about 700 species. Now, a lot of those species are more cosmopolitan. You find them in different types of habitat, not just the salt marsh. So keep that in mind. That's not 700 salt marsh endemic species, but there's a lot of overlap there. The largest order was Coleoptera, the beetles, 36 families and 237 species. And within the Coleoptera, Carabidae, or the ground beetles, was the largest family with 66 different species. And then the most diverse order was flies with 40 families. Here's the breakdown of what we found. As you can see, Coleoptera, it's the largest order to begin with, so it's not surprising that we found the most Coleoptera. Diptera next, Hemiptera, the true bugs, Hymenoptera, the wasps and the ants, Lepidoptera, butterflies and moths, Odonata, dragonflies, damselflies, Orthoptera, the crickets, grasshoppers and such. And then the few orders here are listed down at the bottom, if you can see those. We have the cockroaches, we have the springtails, we have mayflies, stoneflies, caddisflies, lacewings and such. Okay, make up a lot smaller portion of the pie there. So I'm going to get into some of the specific insects here, some of the rarities we have in the salt marsh. I'll try to go through this quickly here. Ischnura barbari is a desert fork-tailed damselfly. There's a colony here. We thought that the insect was extinct. It hadn't been seen since, I believe, the 1930s. Yeah, and there was records from Capitol Beach. So they thought it was extinct, and then it was rediscovered in 1999 in the Whitehead wetlands over there behind the Cracker Barrel on North 27th Street. We went out and we found it also at Capitol Beach, and then the Roper property, which is a private property close to Little Salt Creek South, and Dakota Springs, another property nearby that. And it's a southwestern species, and I think the closest population we have is at Quivira Wildlife area in central Kansas. So it's kind of a neat adjunct population here. Okay, we have a neat little damselfly. The common name is the red damselfly. I like to call it the red velvet damselfly because it has a little velvety, lots of small hairs on it that make it really look like a velvet Elvis painting or something. It's really a cool little dragonfly. It appears to be a connecting link between an eastern and a western species. So taxonomists are unsure whether it's a hybrid between the two, or if it's a new species, or it's just a intergrade, maybe between two subspecies. They don't really know. It used to be collected, well, I think it's been at Capitol Beach in the past, but lately it's been collected at Arbor Lake. I only saw one this year, so it appears to be declining in numbers. Okay, the Salt Creek grasshopper, Trimeratropus salina. This is something that Matt Brust out at Chadron State was really interested in, the grasshoppers in Nebraska. It only occurs here, and then there's a population out in western Nebraska that is possibly a little bit distinct from this. So even though no subspecies have been recognized, this could be a different subspecies. And it's only found at Arbor Lake in the Whitehead wetlands. It was out just a few weeks ago. Here's another bandwing grasshopper that has no common name, Encoptolophus subgracilis. And it was first discovered in Nebraska, I think by Matt in 2006. And it also has a disjunct distribution, populations in Dawes and Sioux counties and the Pine Ridge, and then here in Lancaster County. And it occurs at the salt flats at Arbor Lake. Shoemaker Marsh, and the Whitehead wetlands. I won't even mention the Salt Creek tiger beetle because everybody knows about that, but we have a few other tiger beetles there. This is one, Unota, used to be Cicindela. They changed the genus to Unota now. Togata latulabris, the white cloak tiger beetle. It's a fairly common beetle, but it's very local. And it's only found here. I think this is the only place in the state that we find it. We do find it down in northeast Kansas, about 100 miles away. But this is in the process of being recognized as a separate subspecies from other Cicindela or Unota togata from Kansas and other places. So kind of a unique population here. This is a little scarab beetle, a tiny little thing. It's about 4 millimeters long. It had been only known from 2 specimens that were listed in Scarab Beetles of the Scaraboids in Nebraska in 2008. We found a total of 9 of these, all at Arbor Lake and in Pitfall Traps. And we learned that it's out in the spring and the fall, like some of the tiger beetles are. This is Ripa solidaginus. This is one that we never found. This is a wedge-shaped beetle. They're very seldom seen. Even the more common ones are hardly ever seen. They feed on the flower heads, and you almost have to knock the flower heads and collect them that way. And this is actually this species is a parasite of the alkali bees that are out there. I assume that this is found on sunflowers because of the scientific name solidaginus. We never found this species, but it was named from the eastern Nebraska salt marsh. Get ahead of myself here. Here are 2 stink bugs. Also, we never found these, but there's a researcher named Alfred Wheeler, and he found these at several sites around Lincoln from 2010 to 2014. One of them feeds on saltgrass, I believe, and the other is on cordgrass. So you don't just find them up on the grass, you have to dig around towards the bottom of the plant to find them. And then lastly, we have this little ugly plant hopper. This is Megamelanus bicolor. It has no common name. It's a scarce plant hopper that specializes in saltgrass, and it was first found in Nebraska in 2012, also by Alfred Wheeler. It's also found in the alkaline marshes in western Nebraska, several sites around Lincoln. And then in addition to these, we found 4 species of ground beetles that were new to Nebraska. They're not new species, but they were new to Nebraska. They've never been recorded here before. And then there was, I want to mention, there was a engineering firm, PBS&J. They conducted an aquatic invertebrate survey, several sites along Little Salt Creek in October 2008. Samples were identified by Rithron Associated in 2009, and they found a lot of species that didn't appear in our samples, especially a lot of midges, chironomids. They found using larval keys. The reason I didn't include this data with ours is because some of the sites that they went to were not true saline habitats, and not a lot of salt in some areas of Little Salt Creek. As you probably know, we have these underground springs that provide the salt, and so I didn't want to skew the data any with those. Okay, future research. We need to look more in the aquatic ecosystem. We didn't really do much sampling in the Little Salt Creek itself because we didn't have the keys to a lot of the insects, mostly larval forms that occur in the water, and would have made it really tough. But I think somebody really needs to look into that. And then we didn't incorporate museum records into our database, which could provide diversity trends over time. Like I said, a lot of these insects seem to be kind of disappearing, like the damselfly and some of those things. So I think we could look at it. We know that Salt Creek tiger beetle used to be a lot more common than it is today because the salt marsh was much larger and better habitat. Okay, I'd like to thank some people here. Leon Higley, part of the lab that I worked in, the insect ecology lab. Mary Clausen, Mike Fritz, Tom Malmstrom, Sean Dunn, Bob Harms, Bill Allgaier, Tim Huntington, Tierney Brocious, other undergraduates, workers that helped over the years. And there's this little Salt Creek tiger beetle larva. He's asking if we have any questions.